Good evening, everyone. Welcome to Tamali's classes, and knowledge is for all. In today's class, we'll be talking about proteins, which is again another macromolecule, or you can say biomolecule. In my previous class, I've spoken to you about carbohydrates. Please go back to my carbohydrates class if you have not seen it. So you can go through that and then come back to the proteins class, or you can watch this first and then again go back to the carbohydrate class. So let's start with today's topic. Okay, so first of all, what are proteins? So proteins are basically they are the monomers or they are the polymers of amino acids. So understand that there are amino acids which form proteins. Okay, so basically proteins are formed from 20 different amino acids. 20 different amino acids they align together different structures to form these proteins. Okay, so now how do we know what is the function of a protein? So a function of a protein is also a function of the amino acid present. Whatever amino acids are present, so the function of those amino acids will determine the property of a protein molecule. Okay, so now when these monomers or when these polymers of amino acids when they align together, then they are linked with each other with the help of a bond known as the peptide bonds. So from one amino acid, how are they linked? So in one amino acid, the amino group is linked with another amino acid in the with that carboxylic acid group. Okay, COOH group. So NH2 amino is linked with the COOH group of another amino acid. So there are two amino acids. Suppose in this amino acid NH2 is there and this amino acid COOH. So this NH2 will link with the COOH with the help of peptide bonds. So this is how they will bind to each other or they will link to each other. Okay. So that means what? How can you define proteins? So basically proteins are polypeptides because there are many peptide chains. There are many peptides which are linking together to form proteins. So therefore they are polypeptides. And what are polypeptides? When long chain of amino acids, when they are connected by peptide bonds, then they form this polypeptides or proteins. So going back to the basic amino acid, they will come together and they will form peptides, and peptides will come together and form peptide chains. So many chains will again they will form the polypeptides or also proteins. So if there are two amino acids, they are known as dipeptides, and if there are more than 10 amino acids, they are known as polypeptides. Okay. Now this part first we'll talk about the structure. Here we have similar like carbohydrates. Even here I have drawn a graphical chart where to understand the about the proteins. So here we have the structure, the source, type and class, and the function. So I've told you what is a protein. Okay. So in my next few slides, in my future slides, I'll talk about the sources of protein, how we get protein in our daily life. Then I'll talk about the structure of proteins, the properties of the proteins, then how many types they are divided into proteins, how can you classify proteins, and at the end we'll talk about the importance. So stick to till the end because in the end of the class I'll tell you some very interesting information about proteins. Okay, so now see, these are the sources of proteins. So remember one thing, animals we cannot make proteins, so we cannot synthesize proteins. Okay, this is why we are dependent on other uh, sources of proteins, like we depending on plants. Like plants, they can synthesize amino acids which are making proteins. So we take, what do we take for proteins? We take milk products like paneer, then we take mushroom in vegetables and all. Then we take eggs, we take milk, and mostly animal fats. Okay, animal proteins like fish. Then your meat is there. Okay, so prawns, all these things. There are some vegetables which provide us proteins. So these are the all sources of proteins that we take in our daily lives. Even nuts are also one type of proteins. Okay, when through nuts we get protein. So these are all providing us proteins. Next, we come to structure of proteins. So in we have seen in carbohydrates the structure. Now in proteins the structure is structure of the amino acid. So amino acid structure is which out here you can see the formula below. See, this is the COOH. This one is the alpha carbon because from this carbon is a group attached. Okay, there's an R group attached. This R group can be a hydrogen, it can be a methyl group, or it can be a more complex group. Okay, this R. Then from R, one is your H. Okay. Then again we have NH2, and then we have out here COOH. Okay. So this is the structure of amino acid. You can see the amino acids, it may be polymers or it may be monomers. Okay, and they will form proteins. So the basic structure out here is there is a acidic group, there is a basic group, and there is an organic group. Okay, these three are the basic group structure of the amino acid. Now, it's similarly like we have seen in carbohydrates, even in proteins. Proteins normally they are present in two optically active forms. That is your, it can be L form, or it can be D form. Okay, we have seen in carbohydrates D form, dextro and levo forms. Even in amino acids, similarly in amino acids, all the amino acids except glycine, they have this optically two forms. Okay, that means if the forms which have NH2 on the right hand side, so they will be D forms, and those having NH2 on the left hand side, if NH2 is on the left hand side, then they will form the L form. Okay, and if it's on the right hand side, the D form. Okay, D form of amino acid. Now, this presence, this property to exist in two different forms is known as chirality. Okay, some call it they call it chiral also, but you can say chirality or chirality, whatever your wish. Then amino acids are also amphoteric compounds because they contain both acidic form and they also contain the alkaline form. So the compounds which contain both acidic and alkaline groups, they are also called as amphoteric compounds. Okay. Now, coming to the properties of proteins. What are the properties of proteins? The proteins, they can be colorless and they can be tasteless. Except one, like serine, it's sweet in taste, but other than that, all the other proteins are colorless and tasteless. Okay. Now, if it's a strong solvent, then the proteins are soluble in strong solvent, such as water and glycerin. But if the solvents are less polar, such as ethanol, then the proteins are insoluble in such less polar solvents. Okay. Now, denaturing agents. See, proteins, they can be broken down. They can be denatured down by some agents. Okay. These agents may be physical agents such as temperature, then your heat, okay, pH. But the agents may also be chemically in chemical form such as urea. Okay. Then there are some detergents. So they are all denaturing agents, chemical agents. Okay. Now, there's a term known as isoelectric point. What is isoelectric point? See, this is the point, okay, of a protein in which the pH, okay, the pH out here is 0 because the net charge is 0 out here. Positive and negative both are out here equal, so they are 0 out here, okay. So this is defined as the pH at which the net charge of a protein molecule is 0. So the outer charge will be, so suppose the pi, that means the isoelectric point is like if the pH is below the pi, okay, if the pH value is below the isoelectric point value, then the proteins are positively charged. And if the pH is above the isoelectric point, then the proteins are negatively charged. That means, told you what is isoelectric point? It is the point of a protein in which the pH and okay, which the pH out here, the net charge of a protein is 0. So that will be the pH of the isoelectric point when the charges are 0. So now when this pH is below the pi, then the proteins are positive, and if it's above the pi, the proteins are negative charge. Remember that, don't confuse. Okay. Now next is the molecular weight of a protein. So see, the average molecular weight of proteins, amino acids, is 110 daltons. Da. So suppose we want to calculate the molecular weight of a protein, then you have to multiply the number of amino acids with the average molecular weight of an amino acid. Suppose we have 50 number of amino acids. So 50 into 110 will come around 5500 daltons. Okay. So remember this. Okay. Basically, the molecular weight of protein, it ranges between 5000 to 10 to the power 9 daltons. Remember this because this might come in the exam. The range out here, formula out here, okay, definition of isoelectric point I've given you. So they can ask you that what happens when the pH below the isoelectric point in a protein, then if the proteins are positively, okay? Then what are the physical and chemical agents of body naturation. Coming to the structure of protein, handmade notes that I made. So I have just put it here. Please don't mind the language, the handwriting. So see, structure of proteins. So based on the arrangement of amino acids, the structure of proteins can be of four. Okay. So primary, secondary, tertiary, and quaternary. So looking into the primary part, you can see out here. It's just like a chain type. You can see out here the chain type. So out here, the amino acids are present. The chain type. So on one hand, there is the acidic group, and on one hand, there is the alkyl group, basic group. So, and this is the peptide bonds. See, there are peptide bonds which are binding to amino acids. Okay, the diagram you can see out here, peptide bonds and the amino. So, an example of primary structure of protein is insulin. So, it contains two chains. It is A and B chain. Remember that. So, there are two chains here primary. So, for insulin, next coming to secondary type of structure. So in secondary type of structure, what is happening out here? There are two type of structure. One is the coiled and one is the sheath structure. So coil is also called as alpha helical structure. Okay. So one is alpha helical structure. Number two is beta pleated structure. See, the name itself says alpha helical means it's in a helical shape, or you can say twisted in a right hand shape. Okay. First point, alpha helical is it is twisted in a right hand shape because it's in helical nature, helical structure. Okay. Also, number two is that who first gave the structure, who described it? This is Linus Pauling. Okay, remember that Linus Pauling described this helical structure first. Okay. Then for beta pleated out here, as you have seen in the helical, it's helical shape, right-handed shape twisted. In so beta pleated, pleated means what? Pleats. Okay, pleats. That means the peptide chains they are stretched out till the maximum they can stretch out. Okay, till the maximum they can extend themselves. They will extend and they will stretch out and they will form pleats. Okay, and it will be like side by side. Suppose you are draping a cloth, you can see right this way, this way. It's a line which are parallel to each other side by side. Okay, and this is how they will look. They will look. Okay, so this is the repeated structure. And out remember this, for helical structure there are intramolecular H bonds or hydrogen bonds within the polypeptide chain. But for beta pleated structure, it is intermolecular hydrogen bonds. Okay, link which will link this beta strands. Okay, and for alpha helical, it is the intramolecular hydrogen bond. The intramolecular means is among themselves, okay, among the inside the within the chain. But inter means it will bind the strands, different strands, okay, beta strands. Now, next one is the what are the examples. So in examples we have out here keratin. So myoglobin and hemoglobin, their type of actually hemoglobin is actually quaternary and myoglobin you can say it's a tertiary one. So basically you can take keratin as an example of alpha, and even in hemoglobin and myoglobin, you know, since they are quaternary structures and there are many polypeptide chains linked together, so some of the chains they form a helical like structure. That is why we have given out an example hemoglobin, because the strands out there, the alpha helical strands, they are in a helical structure, in a coil structure. That's why the example out here given hemoglobin, myoglobin, and keratin. And for beta pleated, the example is skin fibers. Okay. Now, okay, see here, you can see out here one thing. You see the hydrogen bonds out here, I've shown here. You can check this one, see. See, this is the hydrogen bonds, see, between among these two. So there are hydrogen bonds, hydrogen bonds, hydrogen bonds like this. And in helical, you can see in helical one like this, like this. Hydrogen bonds are like that. These are the hydrogen bonds. Okay. Moving on to the third one is tertiary. In tertiary structure, you can see that proteins they are interconnected and arranged into loops and bends. So you can see out here in first one we saw alpha helical and your beta pleated. So here you can see the alpha helical is also there inside and also beta plate is there. So out here what are happening? Proteins they're interconnected. Okay, and they're in the form of loops and bends. See, you can see they're bending and they're forming loops. Okay, so in this tertiary structures, okay, there are five type of bonds, okay, which stabilize these structures. What are those bonds? Hydrogen bonds, okay, formed between the O and H or H and N. The ionic bonds are there, which are formed between the polar bonds, okay, charged polar thing, molecules out here. Then disulfide linkages between two sulfur. Then van der Waals force is there. Hydrophobic bonds are between the non-polar molecules out here, the non-polar molecules. So the example out here is myoglobin, and for quaternary we have out here uh, when more than one peptide chains, when more than one peptide chains, when they are interconnected, when they are together, like in hemoglobin. Okay, in hemoglobin the heme protein is there, then your alpha helical is there. Okay, all are coming together. Even the beta heated structure is there. You can see here. Even the alkyl group is there. Okay, not alkyl, it is R group. So all are together. Okay, they're all coming together and forming a quaternary structure. Okay, they're all coming and combining, forming a quaternary structure. What are the bonds present out here? The bonds are just similar to those of the tertiary ones. Hydrogen bonds are here, ionic bonds are here, then disulfide linkages, van der Waals, and hydrophobic. All these bonds are also present in they're also stabilizing the quaternary structures. Now, I would like to mention out here one thing, that sometimes these structures they denature. Okay, so when they denature, they need some proteins, external proteins, which will help them to correct the folding, okay, which will help them to uh, which will assist them in the folding patterns. That means it will help them to from the unfolding, it will help them from unfolding. Suppose there's a stress condition or there is the conditions are not normal, so what will happen, these um, peptides they start denaturing. Okay, it's the chains start denaturing, so that time these external proteins, also known as the chaperonins. Okay, chaperonins are some essential proteins which are helping during these times. Okay, they will assist in the correct folding of the proteins in the cell. Okay, and this is occurring under both normal and stress condition. And this is also not, they will not get involved inside the protein structure, but externally from outside they are helping in this folding, correct folding. Okay. Now, classification of protein. So, based on shape, based on constitution, and based on nature of molecules, proteins can be classified into three types. Okay. First, we'll talk about the shapes. Okay. So, see in shapes here, proteins they can be fibrous in shape or they can be globular in shape. So if it's a fibrous shape, then what are the examples? Actin, myosin, collagen, okay, hair keratin. These are all your fibrous proteins, okay, based on shape. So they are insoluble in water and they are existing in the form of threads. Okay, they are coiled, they remain in coiled position and they are existing in threads to form the fibers. And what are globular? The globular name itself says that they form oval or spherical molecules. The globular, they will form oval or spherical molecules and they are soluble in water. Okay, fibrous are insoluble or globular are soluble in water. What are the examples of globular proteins? Insulin, albumins, and oxytocin hormone. They're all under the globular shape. Okay. Next, coming to the constitution. So based on the constitution, again there are three types: simple, conjugated, and derived. So in simples, what are the example? Globulins are example. They are simple proteins. Okay, simple shape. What are conjugated? Conjugated means when the protein group is attached with a prosthetic group, or you can say it's a non-amino acid substance. When a protein group is attached with a non-amino acid substance or a prosthetic group, then they form the conjugated proteins. Example is lipoproteins. Now you can see that the right hand side out here, I've given some examples. The nucleoproteins, protein plus nucleic acid. Mucoproteins, protein plus carbohydrate. Glycoproteins, proteins plus carbohydrates. Lipoproteins, proteins plus lipids. Chromoproteins, proteins plus colored pigment. Plus chromo means colored, okay? Metalloproteins, proteins plus metal ions. Phosphoproteins, protein plus lipid group. So these are all conjugated proteins. They are all binding together and they're forming conjugates. Okay. Number third is the derived proteins. These are sometimes when proteins they denature or when they are breaking down, they form this derived proteins. Next is the nature of molecules. Based on the nature of proteins, again they can be two types, basic and acidic. So basic means they are existing as cations and they are rich in the basic amino acids such as lysine, arginine. Okay. And for acidic amino acids, we can say acidic proteins, that means they exist as anions and they contain acidic amino acids. That's why the protein is acidic, right? Because they're made up of amino acids. If amino acids are basic, they're basic in form, the proteins. And if the amino acids are acidic, then the proteins are acidic. Okay? Example of acidic proteins out here is blood groups and dipeptide. Okay, what is the importance of these proteins? See, I told you in my previous class that proteins are basically, they help in the repairing process, they help in the damage control process. So basically, suppose when our body is like when a body is sensing some antigens and all, then as we know that antibodies is a type of protein, right? So out here, antibodies are protecting our body, so they have a protective functioning. Then also they have structural support. I told you in my previous class that these proteins are helping in healing, in repairing of tissues, in growth and development. So they're having structural support function. Number three, there are some enzymes or proteins, they act as enzymes or catalysts. That's why they're also known as biological buffers, because they will enhance the rate of chemical reaction. Okay, that's why they're acting as catalyst. Okay, they having this function. Number fourth is that these nucleoproteins, they will carry some genetic characters, so that's why they're helping in the genetic part. They're having a genetic function. Then hormones. Hormones are also some hormones of proteins. So hormones are helping in regulating the muscle mass, then the sex hormones, and for the growth and development. These are all helping in the hormonal part, okay, functioning of the body. Then also there are some proteins in blood plasma. So when suppose the body is undergoing some shock or there is some injuries, during that time this treatment of shock is done um, by these proteins of the blood plasma. So they're also acting as defense and defense. Defense are proteins that are present in human beings which are also having antibiotic property, or they are antibiotic in nature. This is some of the importance of proteins. Now, coming to the most important part, that is the interesting facts of proteins. See, out here, first I give lifespan of proteins. You know that the lifespan of proteins is very, very short-lived. That means the lifespan is very short. You know, every protein that we have, they hardly have a life duration of 2 days or less than 2 days. They can survive for 2 days or less than 2 days. That's why the proteins are very short-lived. Number two is sickle cell anemia. See, in hemoglobin, we know that in hemoglobin is carries, what is hemoglobin? This is the blood component, right? So which is why the blood is red in color. So okay, so hem means iron out here. Okay, so hemoglobin, they are consisting of almost 600 amino acids because they contain two light chain, two heavy chain. Okay, so out here, okay, so they are containing 600 amino acids. Remember, in one chain, what is happening? Since it's forming, this is a single chain. Okay, 150 amino acids, then again 150 amino acids. There are four chains because one chain is two types. Okay, so there are four chains. One chain is 150 amino acids, so total it's 600 amino acids. So remember, in a chain of a hemoglobin, there is a amino acid known as glutamic acid. Sometimes what happens, this glutamic acid is replaced by valine. Okay, valine. So in a hemoglobin structure, even if one amino acid changes, it will lead to a change in the functioning in the property of the uh, protein. Okay, so if glutamic acid is replaced by valine in hemoglobin, okay, it will lead to sickle cell hemoglobin. This is a type of disorder. Sickle cell disease is also a disease, sickle cell disease. Okay, so this is sickle cell anemia. You have heard about sickle cell anemia. So this leads to sickle cell anemia if there is a um, change in even one amino acid. In this case, glutamic acid is changed to valine, leading to formation of sickle cell anemia. Number three is denaturation of protein. So we know that egg is a protein, right? We say that egg gives us it's a protein source because in egg it contains white part known as albumin. It's a protein part, okay, in the egg. So what happens? You have seen when we toast egg, or when we fry the egg, what happens when the white part, it will solidify, it will precipitate, and it will form a, you can see the color changes. Okay, the color change, the transparent part changes into a white part, or you can say it's a bit creamy color type. Okay, so what is happening out here? The protein is denatured. Okay, so there's a denaturation of protein. The albumin protein is denatured. Okay, and since it contains hydrophobic groups in the outside part, what happens? when the boiling occurs, then these groups what happen? They will start aggregating. Okay, they will aggregate, will precipitate, and they will form that white structure, or you can say the solid structure. It is a precipitate like formation. Okay, they will aggregate. So this is all happens. That means the protein has been denatured in the egg part. Remember that why we are getting toast, why we get to the white part, because of denaturation of the protein part of the egg known as albumin. Okay, number three, very very interesting. So there's a protein known as picachurin and also sonic hedgehog. So you have heard about Pokemon, right? You have heard about Pokemon Pikachu, the cartoons, also Sonic the Hedgehog. So there are some proteins which are named after these cartoon characters, okay? Like Pikachurin and Sonic Hedgehog. There are two proteins which are named after these cartoon characters of Pikachu and Sonic the Hedgehog. This is again one interesting thing to know. Also, in our hair, we contain keratin protein. Okay, so human hair we contain keratin protein. Now, this keratin there are sulfur bonds. So, and okay, you have seen some people have curly hair, some have straight hair. So since our hair contains keratin protein and keratin protein contains sulfur bonds, so the more amount of sulfur links, okay, the more amount of sulfur links, the curlier the hair gets. Okay, suppose if we say that person's hair is too curly, then we can say that the keratin present in the hair, it contains many sulfur links. Okay, it contains many sulfur links. Okay, these are some of the interesting facts about proteins. I hope you have liked the class. So if you have liked it, do like the class, and if you have not yet subscribed, do subscribe my channel for more such videos on biology, and do give me suggestions and let me know what else you would want to learn in biology subject. So have a great day, and thank you for watching the video.